Hello everyone, I'm Steven and you're watching Steven Loves Science. Now, we've talked a lot about DNA on the show before. We've talked about its replication, its transcription, its packaging, and last summer we even took a little field trip to Carlsbad, California, where we visited Life Technologies and learned about their sequencing platform, the Ion Torrent. But we've never really talked about how DNA is relevant to you. Well, I mean, of course it's relevant to you, but how is it relevant to you on a more personal level? So the year 2003 marked the completion of the Human Genome Project, which was a 13-year-long government-funded effort to completely sequence the human genome. And it wasn't the only one going on. There was also a private one going on. They were kind of in competition. Who won? No one really knows. It was kind of a photo finish, but we're all winners because they sequenced the human genome. But the government-funded effort cost an estimated $3 billion. And that was 11 years ago. So the other day, I went online and I picked up the 23andMe Welcome to You Personal Genomics Genome Whatever Kit. So this is the only one out there. There are lots of others. You can buy one from National Geographic, whatever. So the point is I got this for $99. So what really can this information tell you? So the 23andMe company has two main hooks. That's going to tell you its genetic ancestry, which others can. National Geographic has one, and your potential disease risks. Well, it can't really say that anymore. It used to say that, but then it got into a scruffle with the FDA, so now you can't really say that. But we'll talk about that as we continue. So all of these genotyping, personal genotyping kits work on the same principle. So we need to understand what they're looking at when they're looking for your genetic ancestry and disease risk. So we've talked before about how DNA is made up of these 4 different chemical letters, right? A, T, C, and G. So let's consider for a moment a sequence of DNA from our reference genome. So here, according to our reference genome at this position, we have A, T, C, A, and T. Now, of course, not everyone is going to necessarily have exactly those letters, because there's variation that exists in a population. This is very important for evolution, which is going to be in our next video. So here, not everyone is going to have exactly A, T, C, A, T. So to emphasize this, here we have James. And James, oh boy, James, he is just always a troublemaker. Instead of having a C, he has a G, right? And there are a few possibilities that can happen, right? The first is this stretch of DNA, it's junk. It's non-coding DNA, no effect on any of James's life processes. He is just fine. He can have whatever letter he wants. He could have X, Y, Z, O. Who cares? He's fine. That's non-coding DNA, whatever. Next situation, let's say I'm oversimplifying here. This is a gene encoding for hair color. So normally you have the C and you have really jet black, dark hair. But now he has a G, so maybe he has light blonde, curly hair. And that actually could probably affect his biological fitness, his reproductive fitness from a sexual selection standpoint. It'd be interesting to see if that blonde curly hair is really attractive with the ladies, so maybe we'll see the frequency of that allele spike up in the population, a lot of James Jr.'s happening in that population. And now a third situation, third scenario here. This stretch of DNA, this codes for super important gene number 1, super important protein number 1. I'm telling you, if you don't have that C here, super important protein number 1 is not going to work. You're screwed if super important protein number 1 doesn't work. And there are a lot of genetic diseases, what are called monogenic diseases, such as Huntington's disease, muscular dystrophy, Fragile X syndrome, that are all caused by a single nucleotide substitution, which subsequently causes an amino acid substitution, which affects the functioning of the protein. So if James has a G instead of a C, he's going to be swimming with the fishes, and Jaws, Paws, the cat shark, is going to destroy him and just eat his dead body and just tear it apart. So let's get into what you really came here for, right? You want to learn about what, for $99, won almost an entire Benjamin, and I guess other currencies if you're an international viewer, can this kit tell me about myself? So let's first get started with talking about disease risk, because we already kind of went into it when we were talking about James's super deleterious mutation in his super important protein 1. So for diseases that are monogenic, I can tell you if it's a dominant disease, meaning you only need one copy to have the disease, that if you have a G instead of a C, I can tell you with basically 100% certainty that you will have this disease. But unfortunately for the scientists studying them, most diseases are not as simple as monogenic diseases like Huntington's and muscular dystrophy when looking at them from a genetic aspect. So diseases that are really high up there in the causes of death, cardiovascular disease, cancer, are what are called polygenic multifactorial traits, multifactorial diseases. So as the name may imply, it means that multiple factors, both genetic and environmental, are going to play significant roles in your disease outcome. So why did FDA get into this scruffle here when talking about disease risk? Because they wanted to say if you have this SNP, then you have a 27.896% higher chance likelihood of developing this disease than someone who does not. But we cannot really say that at all. Scientists are still struggling to understand the relationship that these genes play with the environment in tandem to ultimately have disease outcome. Now, a lot of you may have heard about, through the media, a gene, I guess two of them, called BRCA1 and 2, which have shown strong associations with breast cancer in scientific literature. So BRCA1 and BRCA2 are code for genes that are tumor suppressor proteins, I believe. And of course, as you might imagine, having tumor suppressor proteins is very important, making sure they work properly. But if you have certain mutations in those genes, in BRCA1 and 2, then you have an increased likelihood of developing breast cancer. But those are really only two genes. So we know that many genes are involved in these pathways that can lead to cancer or other diseases. And we know that the environment also plays a significant factor. So you could have every single gene that is known, I'm exaggerating here, that is known to influence heart disease. But if you go out and exercise daily and eat right, you might never develop heart disease in your entire life. So really, the environment does play a significant role, and especially in diseases like cancer, much more so than genetics. I'm over-exaggerating as well, but the main covariates are influencing factor that plays the most significant role is smoking. Smoking's way up here for the most part. For the most part, I'm lying to you a little bit, but for the most part, yes, smoking is going to be way up here. And genetics, all the way down the floor. Here is smoking, all the way up here. For heart disease, genetics certainly do play a role. It's currently our scientific understanding. But ethanol use, if you're using ethanol on an excessive and regular basis, ethanol, drinking alcohol, then you're also putting yourself at a higher risk of developing these diseases. And of course, you could drink a lot of ethanol daily, gallons, and maybe not ever develop heart disease. So we're still trying to understand how these genetic and environmental factors play together to ultimately determine disease outcome. And we'll most likely never understand fully how the genetics and environment work together. So now let's get to genetic ancestry, which is a bit more complicated to understand. So here we have James. And let's suppose James here is a resident of the South American continent here. Actually, let's break this bridge here. I'm rearranging the globe to make my explanations make more sense. So here we go. Here we have James, who has his gene mutation, and that gave him really nice blonde hair. And lo and behold, the ladies in South America love James's blonde hair. So James is biological fitness is going to skyrocket, and we're going to start seeing a lot more people, a lot James Juniors are going to have that G allele, and eventually we'll see that basically the frequency of this G allele is sky high, and we can tell based on whether or not an individual has his G allele. That could be a determining factor in evaluating whether or not they are of South American descent. So these are called AIMS. These SNPs that have to do with ancestry are called AIMS, or ancestry informative markers, and they arise from geographic isolation and sexual selection. So they arise from geographic isolation because if we start off with some people with G's here, and we're not going to introduce any new alleles, because in order to introduce new alleles, we have to have people from coming elsewhere coming into this population and mating, thus propagating their alleles. This is called genetic admixture. But if it's isolated, and of course I just destroyed this land bridge here, then there's going to be no one coming, so no one's going to introduce their genetic material, and we're going to have sexual selection because, of course, the ladies love James's flowing blonde hair, so they want to reproduce with him and people that look like him, and therefore we see an increase in the frequency of certain alleles that influence hair color, and of course there are other alleles in populations that we see are of higher frequency across ethnicities. So that's how the genetic ancestry component works. And another thing I wanted to mention, I didn't put it on the board, that at least I know 23andMe does, and I promise I'm not a spokesperson for this company. They have a lot of flaws with them, and I'm not a spokesperson by any means, but as 23andMe genotypes you, they will collect all your information in a database. And we know, obviously, that siblings and other relatives, whether it be a first degree, second degree, third degree relative, you will share a much higher proportion of your SNPs with them than you will with someone of the general population. So if you get genotyped with 23andMe and your long-lost brother or cousin gets genotyped with 23andMe, then you'll both be in their database, and 23andMe will be able to match you up and say, hey, your long-lost cousin just got genotyped with 23andMe, go meet up somewhere, get a coffee, you have catching up to do. So that's pretty much the gist. I did want to get into talking about how genotyping works, but I think that's too much of a mouthful for today. I'll let you guys digest, so we'll actually talk about this next week, and I'll post my evolution video 2 weeks from now. So as always, thank you very much for watching this episode of Stephen Loves Science. So to answer the question, is this worth $99? I think so. If you have $99 to spare, certainly. It'll tell you a lot about yourself. Of course, for the disease risk, you'll now only get raw genetic information, which is saying your genotype, which, unless you're a geneticist, you might have some difficulty understanding. So your doctor probably will have difficulty understanding also, but I don't know, it's neat. I think the genetic ancestry component is the coolest. So please join me again next week, and don't forget to subscribe, rate, and comment.